Great. Thanks a lot, Vijay. I mean, I thought you ran that panel just absolutely beautifully, and hopefully we can do the same in panel number 2. And so um, here are our panelists, but let me give a short introduction and try to refocus us on what we're going to talk about within this panel. And I think here we're going to dwell a lot more on drug development tools, right? Step a little bit away from the clinical practice aspect of biomarkers for IBD. I think one of the things we have to remember is in order to have regulators accept the data that we produce, they have to see that data, right? In order to make decisions around whether those biomarkers are valid or not. You know, interestingly, in the last panel, we talked a lot about real-world data or natural history type studies to help drive biomarker discovery and possibly even biomarker qualification, right? But one thing we didn't dive into is how do we leverage the randomized clinical trials that are being done by drug developers, and can that be used to help drive some of our biomarker qualifications? I think the answer there is yes, absolutely yes. I think what we also have an opportunity within this panel session to do is to really figure out how we partner with health authorities as drug developers, as academic researchers, to, on a scientific level, and create the focus that we need. Right? Because I heard a lot of different ideas during the last panel session and in the different presentations, but I think we're missing that focus piece. I think Stefan did a good job in talking about the systems biology and how that can bring focus. But I think one of the things that we definitely have to talk about is where do we start and really what's our goal at the end? And I know everybody wants a surrogate biomarker, right? But what are the intermediate steps to get us there, to be able to get the biomarkers that we want and need within drug development? And so I think what that comes down to is a community focus, because it takes a long time to work these biomarkers through the regulatory process. So that's something to consider, right? And so, you know, with that, let's treat this as an opportunity, right? To partner as a community and to understand from both the drug developer side and from a health authority side, what's really needed out there, right? What are these key biomarkers that can really transform the creation of therapies for Crohn's disease, ulcerative colitis, and other IBD type activities? So with that, I'm going to call on my panel, and what I'd like you to do is to go ahead and begin to introduce yourself. So why don't we start with Tara? Are you on mute, Tara? I can see you there.
Sorry, multiple mutes. I was trying to fix the camera. 1 second, I apologize.
No, take your time, it's okay.
Okay. Hi, so good morning, my name is Tara Altepeter. I'm a pediatric gastroenterologist by training, and I work at FDA in the Office of New Drugs in the Division of Gastroenterology, and really IBD is my love and my focus. I probably spend, 50 to 70% of my time doing just that. And so I do have a lot of opportunities to see, from the commercial side of things, the work that is going on with biomarkers, and, many areas in which they have been helpful or, could be more helpful in the future. So thanks for having me.
Thanks, Tara. Elizabeth, do you want to give a quick introduction?
All right, if I can unmute myself, can you hear me?
Yes, we can hear you.
My name is Elizabeth Amstutz. I'm heading up the EUCON Regulatory Affairs team in GI Neuroscience in Takeda. I've also been involved in 3TR regulatory work stream and really excited to be here. I have over 15 years of regulatory affairs experience.
Okay, great. Well, we're going to use that 15 years on this panel. So everyone knows Fred. So Fred, you don't have to give an introduction, but we look forward to your comments on the panel. Pam, do you want to give a short introduction?
Thank you. Good afternoon. I'm participating from Takeda's Global Regulatory Affairs Gastroenterology Group. And I'm very pleased to be here and contribute. Obviously, my career's been focused on drug development, most recently in gastroenterology. And I do want to just take a minute to pick up, John Michael, you commented on, a community focus here as the task is complex and potentially lengthy. And I thought that was a very nice way to set the stage. So wanted to comment on that. Thank you.
Thanks, Pam. Chris, would you like to give an introduction? I think you're on mute, Chris. I'm sorry.
Okay. Now can people hear me?
Yes.
Excellent. Thanks, everyone. Just to be clear up front, I am not a gastroenterologist, but I have friends that are. And that aside, when I started at the FDA, I actually was in the GI division looking at immunomodulators for inflammatory bowel disease. My PhD is in immunology, so it was a good fit. More recently, I'm the director of the Biomarker Qualification Program at FDA within the Center for Drugs Evaluation and Research. And certainly this particular panel discussion around biomarkers and how we look at them as regulators is very much what we work with all of our clinical colleagues within the Office of New Drugs to ultimately bring biomarkers forward that are regulatory ready for a particular context of use. And certainly the panel one discussion touched on a number of topics that I think are relevant to that and looking forward to the conversation.
Thanks, Chris. Ricard, would you like to give an introduction, please?
Hello, my name is Richard Vesely, and I represent EMA on this panel, but actually until 2009, I worked in Slovakia as a pediatric rheumatologist. So I'm a physician by the background, so I am actually wearing both hats that might be useful for this community approach to the discussion. However, in this discussion, I will represent regulators. I started in 2009 in EMA as pediatric coordinator. Later, I joined the rheumatology, immunology, gastroenterology office as head of office, and recently, it's exactly 1 year now when I joined the COVID-19 team. That's basically my background. I'm looking forward to the discussion. There are a number of my colleagues attending this meeting. I don't know if they will be able to join the discussion if needed, but I believe they can at least enter the chat box. So looking forward to the discussion.
Thanks, Ricard. So yeah, that was an excellent reminder. So we have that Q&A box. So please type your questions in there. I saw there were a couple questions also in the chat box, and we'll circle around to some of those. So I think this is a great opportunity now to really interact with health authorities around these questions. But I think one thing to remember, right? This is a scientific conversation. This is not a formal, regulatory conversation where these where these points are going to be binding. We're all speaking as scientists, and keep that in mind. Likewise, the folks representing pharmaceutical companies, same thing, right? They're bringing up their scientific opinion and trying to figure out solutions to these different questions. And so, this is a good forum for scientific conversation, and let's realize that as participants. And so where I'd like to start off as the lead moderator is really with you, Tara, and in your presentation. Could you, share with those who haven't had an opportunity to go through your presentation, where you really see the opportunities for biomarkers within the IBD space?
Sure. So in the presentation, I think I outlined about 4 different potential areas where we see biomarkers might really be useful in moving the field forward. Can you guys hear me okay?
Yes.
Okay. You know, some of the, I guess I can start with the ones that maybe have more of a regulatory focus. You know, the first, which I think is really important and potentially impactful, is in the aid to dose selection. So from a scientific perspective, one of the challenges and trends that I've observed over the last several years, you know, as the space really becomes a lot more competitive, is more and more rushing, right? Companies want to get to the finish line faster, and the idea of doing phase 1, 2, 2B, 3 programs that take a decade is, you know, maybe less practical than it was in the past. And so one of the things that sometimes gets lost there, though, is really robust dose selection and characterization of exposure-response relationships, which, you know, not only has potential risk to the phase 3 program, but, you know, I'm a pediatrician at heart, and so one of the areas that we really then struggle with is in getting to the step of moving programs to pediatric development and not having that really good characterization and support for the initially selected doses in adults really limits the degree of extrapolation and our confidence in relying on those adult data for the pediatric program. So one area that I do think there's a lot of room to apply biomarkers would be as an aid to dose selection. You know, we've seen a whole variety of them used in that context, and as we're getting into newer, more unique mechanisms of action, you know, sometimes there are opportunities for an individual firm to have a biomarker that's very specific to that mechanism of action. And so it may not be something that's, you know, widely used and commercially available, but might be helpful in that context. Some of the other things, just briefly, that I outlined in the talk in terms of regulatory use, you know, safety biomarkers are always a welcome addition, be it the ones that, you know, we use all the time in terms of measuring or detecting toxicity, you know, things like creatinine or liver enzyme tests, things like that have been around forever, you know, but also a real desire to look to the future and think about where there are ways to potentially predict toxicity to help make sure that patients do as well as they can with the therapies that are being developed. So from the regulatory side, I think those are a few of the hot topics. And then the other issue that I talked about a little bit towards the end of my talk was the idea of things that could be used to help in selecting a subgroup of patients that might best respond to a given mechanism of action. And, you know, maybe there's not as much use for that in the regulatory context, but I think, you know, over the next decade, as we get, you know, a good problem of having more different efficacious drugs out there, as practitioners, we're really going to be faced with trying to think about, you know, which one should we try first, or how are we going to select the best drug for the specific patient in front of us? And, you know, one of the tough parts about this from the regulatory side is it's rare that we can require a comparative trial, right? So once in a blue moon, a company chooses to do that, but that isn't typically something that we require, you know, in the standard course of business. And so, you know, there may not be those great prospective head-to-head trials where the data will help to inform a clinician in a really robust way as to which one is the best or which one is the safest. And so in that context, I think there is a lot of opportunity for us as a community to start to understand more all the different phenotypes of IBD that really are out there, and are there subgroups that really might do better with one mechanism of action versus another. But you know, that has to start being thought about way in advance, right? Those are the biomarkers that may be drug specific that an individual firm might start to develop and use throughout their development program. And then, you know, subsequently would have to have an interest in developing, if not as a companion diagnostic, then, you know, maybe as a commercially available assay down the line, but to try to help bring that focus towards really individualized medicine.
Thanks, Tara. I think that was a great overview of your talk. Ricard, do you have anything to add to that from an EMA perspective and the type of activities that you are involved in?
Yeah. So first, thank you for including us to this discussion as the European regulator. And Yeah, I followed very carefully the discussion of the first panel, and also from the presentation of being the patient representative. I can say there are very different expectations about biomarkers. And you said it in your introduction to the webinar that there is a big difference between clinical expectations and regulatory application of the research of biomarkers. So from my regulatory point of view, what we can deal with is the requests for label changes, actually. So what biomarkers can be used for labeling of medicines to determine the benefit risk of the drug or the other values of biomarkers, like context of use, like a safety biomarker or prognostic biomarker, etc. And I heard with great interest the comment that there is a passage about predictive biomarkers, which is actually one thing that is, from regulatory point of view, very important, actually for labeling, and also the comment about the pathophysiology. And actually, IBD is an example of a disease like rheumatoid arthritis or other diseases, which is actually a group of diseases. So understanding better the pathophysiology or the biologic background of the disease would help a lot with labeling, because when we know which biomarker actually reflects the pathophysiology of the disease and actually has a predictive value in that sense, would actually very directly lead to the labeling change. So in this sense, I have to say that we as regulators are more reactive. We listen and follow the requests from developers of medicines and biomarkers, but we usually are not part of development of biomarkers. And we can discuss later what procedures we have for this dialogue. But we are not expected somehow to be part of the development of biomarkers. Maybe this is a good forum to discuss that, whether we should be somewhere included in the development of biomarkers. And maybe as an example, we could listen from 3TR representatives there, where in the presentation there was a work package 10 focused on this regulatory acceptance. So what is actually expected as a work in this work package, and how we can interact at that level. So maybe this is for this moment from my side. Thank you.
Great, thanks, Ricardo. I think that's really helpful. So, Chris, I mean, you basically, take all this science in your role and then work with requesters, biomarker folks, and try to get this all to work, right? To where, we can bring these biomarkers forward and achieve regulatory endorsement. I think we covered a number of things this morning as far as the science behind this. But, what's your feeling on the state of where we're at with these IBD biomarkers, and how can we start thinking about moving forward? You and I have had plenty of conversations around these biomarkers, but could you share your thoughts around that, Chris?
Sure. I mean, one of the things that we as biomarker developers for regulatory purposes do as part of a day-to-day, work is to work with external groups as well as our internal subject matter experts to look at the science of the proposal and to see whether or not there are knowledge gaps and how we might be able to fill them to get towards an ultimate goal, what we call the context of use. Thankfully, since the 21st Century Cures passed at the end of 2016, We now have a mandated addition to our Code of Federal Regulations that also includes a number of provisions that have really proven to be quite helpful around biomarker development. The main one is around the transparency provisions piece. So both with cures and as part of our PDUFA 6 negotiations, FDA has been front and center around participating actively and collaboratively with external groups around not only the idea, but also the science to support it. And that information is then shared as part of a public resource on our website. So you get the benefit of seeing what proposals are being developed. Information about those proposals is made public. And then likewise, FDA did an additional step where we made public our decisions around accepting or not accepting a given submission, but then also made public what recommendations we shared with that party or parties externally around continued development. So it has really led to a shared learning culture where everyone can see what's being proposed, what we think about it, and then incorporate those ideas and suggestions into their own interests and goals. So if you're developing an imaging biomarker, certainly look at what we've said around other imaging biomarkers and bring that into your project plan. And within a relatively short number of years, we've seen the quality of our submissions grow. We've seen that there's been a communal understanding of the science to support biomarkers for regulatory use. And I think that will only continue to do so in the future. Around IBD specifically, I mean, certainly Tara sees this day in and day out much more than I do. What we have seen over the years that I've participated in different types of IBD conferences is that there's a lot of ideas, but as much as we kind of encourage those ideas to be brought forward independent of a drug-specific program, namely engage with us around a qualification effort, so far we haven't seen as much activity as we certainly could. So I would encourage this group, as I've encouraged others, bring your ideas forward, work together collaboratively, share insights and ideas, and see where you think the science tells us, and bring that forward as part of a package around what you want as a goal, because we'd love to be able to work with you. The last thing we want is people to continue to stay in their silos externally, do their efforts, spend their resources, spend their time, and then engage with us at the end. And by then, it's usually too late. We want to be a part of the process early and often.
Thanks, Chris. That was some great advice. So what I'd like to do is I'd like to pivot from that conversation right there. And Ricard, you basically, teed up my next question, and that is really to the 3TR folks, right? Elizabeth, Fred, Pam, about Work Package 10. And what are these different ideas that you're bringing forward, and how, can you talk about some of those ideas, and then how you'd like to engage with regulators to go ahead and make these ideas real. And Vijay, please also join in here.
I can start actually before Elizabeth. So one of the things that when we decided on the work packages, regulatory engagement became a priority early on, and so from the get-go. So we never said it's like, we were not going to do this 3 years later and say, hey, by the way, we got these results. So I think that was the biggest change. And I think maybe Elizabeth, you should take it on from here, but I think that was a very critical JST meeting early on, even before the meeting.
Yeah, no, Vijay, I absolutely agree. So from my perspective, John Michael, the fact that we're sitting down with Chris, with Richard, with Tara, and having this discussion, and 3TR is a great achievement as is, and it's highlighting the need of the initiatives of the AMI to start this dialogue and not only focus on Europe, but look outside and bring the convergence together. So from my perspective, there's a lot that we can do within the project, and as we go through and raise the awareness of what are the regulatory requirements for qualification of biomarkers, how do we want to combine the strategy and put it all together, we will start having much more of these dialogues across with FDA, with EMA, and hopefully jointly as well.
Great. Fred, Pam, anything to add to that?
I think on a scientific level, right? So 3TR is a very ambitious project, multi-year project, where the good news is, I think, that the scientific dimensions we are collecting in particular in IBD is quite remarkable, and we have great hope that that synergy of approaches and focuses, microbiome, transcriptomics, proteomics, etc., will boost the discoveries of those biomarkers you highlighted, right? Where you can optimize response, you can understand non-response, you can better understand how you would measure those response, etc. And then I second the remark around, we absolutely wanted to engage the health authorities early and have the conversation, as it was highlighted by Chris, to have that as a common conversation among us, and how to get to the ultimate goal, which is qualification of some of those biomarkers, if even one, so it can be used in any one of those applications I just mentioned. It's an ambitious project, but we're optimistic and hopeful.
So, Michael, can I add a comment there, if you don't mind?
Yeah, please.
So one of the things that you know, we're always telling people to engage with us early. Let me just kind of give you, you know, a 30-second of what we're looking for a letter of intent, because I think sometimes people misconstrue that the letter of intent means that you have to have data and data sets and have all this information. Where in fact a letter of intent is primarily focused on a couple of different features of a proposal. The first is the drug development need. You know, clearly define what is the need that you're looking at, and please be sure that it's a drug development need. You know, there are other needs by other communities, clinical and so forth, but we focus on drug development. The second is craft a context of use based on that need. And then, you know, we want to know which biomarker or biomarkers are you targeting. It doesn't mean that if you're doing a panel, for instance, of multiple different biomarker analytes, that you have to have the perfect signature. Just tell us what you think is biologically plausible based on current understanding, and that, you know, you're going to be looking at those numbers and refining them over time. That's completely okay. And then the final piece that we look at is, what are the measurement platforms for those individual biomarkers? Is this something that's commercially available? Is this something that you've developed in, you know, a research environment? Or is it just an idea? You know, we certainly don't want the idea piece. We want there to be some level that this could in fact be developed. But that's it. It's not a data exercise. You don't submit data sets to us. So as you can see, basically if you have a sound idea based on a targeted need and can give some evidence to support what you're proposing, that's what you need to have our first conversation. So I just wanted to make that clear, because I think there's a little bit of a misunderstanding of what you need for a letter of intent.
Thank you, Chris.
Yeah, thanks for lowering that hurdle, Chris. Oh, go ahead, please.
Jump in and ask the question. What Richard highlighted is the need for, and the patient perspective brings that loud and clear as well, the need of prognostic biomarkers that can influence the label. So does that fit within the drug development umbrella, and how to structure that? Because I think that's one of the challenges from the FDA perspective of from what we're seeing currently, right?
Yeah, I can start, and then Tara, I'm sure you have a perspective as well. So patient perspective, exceedingly important, and biomarkers can help or enable to ensure that patients' needs are being met. So in the example that you gave around prognostic biomarkers, you know, patients with a given disease or condition want to know, what is the likelihood that that disease or condition is going to worsen over time? And will this particular therapy help to change that trajectory, or even slow or reverse it? And so prognostic biomarkers can be really helpful for that purpose. And certainly we have context of use in that space. So that's a very legitimate and commonly a common target that people use around prognostic enrichment for clinical trial design to get at that very question. And then Tara, I don't know if you wanted to add to that.
I think to get at the idea of what can make it into the label at the time of initial approval really depends in part on how available a given biomarker may be, obviously in addition to our understanding of its you know, parameters and how it performs. But, you know, outside of that, the information that we put into the label, we want people to be able to use it. And so, like, I'll give a couple of specific examples, right? So if we, for example, saw a trial conducted, and I'll just pick CRP as the example that was brought up earlier, which is widely available, right? So if we actually got a trial that was specifically enriched to only enroll patients with a very high level of CRP, just as an example, and that turned out to be important, and, you know, that's something that you would want people to know, that it, you know, it works in this population who may be a little bit different or a little bit more refractory than others. You know, it's possible that something like that could make it into the label, at least in the description of the clinical trial in section 14 for our purposes. But by the flip side, if it's something very novel that's proprietary or that's specific to a given drug and it's not commercially available yet, you know, I would think probably not. So part of it, I think, is sort of thinking about it more holistically, you know, not only, you know, is it fit for purpose? Does it perform well? Does it meet our needs? But then also that next step in, you know, we really try to focus on keeping our labels succinct, which I guess is a little bit laughable to say that because they're still very long, but, you know, we're working towards trying to make them succinct and helpful to the physician. And so we want to put information in there that's going to be actionable. So I think that's the other side of that, right, is to think about, you know, I don't envision us labeling something that's not widely available, in addition to sort of meeting the needs in terms of the validity aspect.
Thanks, Tara. Ricard, do you have any points you'd like to make?
Just to add to this patient perspective, so some of the wishes that being expressed was that he would appreciate having biomarkers that would reflect better and in real time how he is feeling, or predicting how he will feel in 2 weeks. So this is a clinical request. This is a clinical question. For when it comes to our area, it is whether this can be influenced by medicines. And so that's what I meant when I said not every request for biomarkers is actually addressed to the drug development. And that should be distinguished as well. Although I understand it is absolutely important and necessary to develop biomarkers for clinical use, but not all biomarkers are actually for drug development use.
We follow up on that point from Ricard. We have tremendous patient engagement in 3TR, and we proud ourselves from having patient perspective to some of the research we want to conduct, and the inclusion of patient-reported outcomes and patient voices is very important. So to your point, Ricard, not everything is focused on drug development, but also what benefits patients and what is meaningful to them in terms of their symptoms. And I think the other notion I wanted to bring up in 3TR, and now I'm working as an advertiser for the consortia, but while today we're focused on IBD, we certainly have other diseases we're looking into, and we actually want to use those different diseases to inform each other. There's commonality and there's differences, but it would be remarkable, at least as a scientific idea, and the conversation is based on science, that we can leverage learnings from disease A for disease B, or even use a biomarker in disease A in disease B, and so on. So that's another notion we're going after. And now back to patients, the fact that we included a common patient-reported outcome across all of those diseases, again, highlights how we're focused and sensitive to patient voices, and not ignoring some of the symptoms which otherwise are not as addressed as they should be. And just to give an example, we've decided to, for example, focus on fatigue, which is one of the classical symptoms all of those immune-mediated inflammatory diseases have. So, yeah.
Just one more thing I think I would just say is like for microbiome, and I'm a spokesperson for microbiome. So Stefan and I have been pushing that across every disease. So even if you have MS, RA, lupus, COPD, we're going to study baseline microbiome so we can get that discriminant function, right, as we're saying with Fred. So Stefan, do you want to add anything else in here? Or you're welcome to say something about the regulatory interactions planning.
One additional comment, if that's okay. So, you know, the comment that was made about, you know, the importance of prognostic measures and sort of incorporating the desire of the patient to predict or understand how they may do differently. I just wanted to also make the point that though it may not help your drug development program proceed right in that moment, I do think that there are really unique opportunities for the individual firms to start to collect some of that data to help in the development of biomarkers that will be used for that purpose, you know, maybe 5 years after approval. So one of the things that I do note is that although sometimes we get great proposals from academic groups and consortia to try to do some of this work, it's really hard for them to fund and conduct and run a trial that might have 4 or 5 years duration of patient follow-up. And at times that's really some of the information that's needed if you're really interested in, like, okay, you know, the 52-week trial that you do to get your drug approved is going to at least tell you that it's making people better in the relatively short term, right? That's sort of the minimum that we require. But if you're really interested in altering the natural history for that patient and getting out of this setting that many people find themselves in, where you start a biologic or drug and it works for 6 to 9 months, but we know that a large proportion of patients switch therapies within 1 year to 2 years of starting them, and we want to instead be able to see, you know, we think that this medicine is going to work for you and keep you well in the long run, you need to have that longitudinal data, and it can be very difficult for, you know, individual or small academic groups to be able to fund a study for that time frame. And so I think that, you know, in so much as that people are able to work together or at least start to collect some of that data, you know, many of our trials, the first patients that join do end up in that extension trial for 3 or 4 years. And so that is really valuable data and, you know, perhaps isn't being harnessed and shared in the ways that it could be to answer some of those questions that are important to patients down the line, even if they don't necessarily impact what would come in that initial drug submission.
Yeah, I think this is really where we see this sense of the community. And thank you for making those points. I think very important. And, you know, obviously, strictly from a drug development standpoint, data are all standardized now. So there is that opportunity to be mindful as data are being collected that over the long term, this could contribute ultimately to identifying, you know, a biomarker that would potentially, you know, be very helpful either to other products coming along later or as a prognostic factor and things of that nature. So I think this is really where this concept of community comes in, that we're all contributing to a common set of data.
Pam, I think you bring up a great point. I mean, so if we look at, you know, randomized controlled trials, of course, the gold standard is being measured, right? And so if indeed there are exploratory endpoints, exploratory biomarkers that are included, it's a perfect opportunity to go ahead and make that comparison and understand that those exploratory biomarkers can add to, you know, not only internal decision making, but then regulatory decision making around that particular therapy, maybe around that particular disease.
And Pamela and John Michael, like to add to that, certainly we're always trying to increase our scientific understanding. And even if you're doing it in an exploratory fashion, that has the ability to contribute to that knowledge. But it only is able to do that if you provide the documentation to support what you did. So gathering information around biomarkers and then not including the units or the measurement platforms that were employed or the protocols by which the biomarker was collected, all of that, if you can include that and share it with FDA, we can then look across programs and do more of a meta survey to see whether or not that biomarker has potential value. But as companies gather this information and kind of do their own analysis, many times the data is, you know, more limited, and there's limits to how much we can say based on that single study. And in the absence of the things that I mentioned, it's really difficult, if not impossible, for us to then look across data across studies. So please share as much of the details of how you did your data collection as part of your information that you share with us. That would be exceedingly helpful.
Let me give the c-path commercial. I mean, we heard a 3TR commercial. I mean, that's exactly what we do. When we pull our public-private partnerships together, our consortia, we're looking for different pharmaceutical firms to go ahead and provide this information, right? And we've worked with Fda, Ema enough that we know what they're going to want, right? And so we can help guide that regulatory approach towards the endorsement of these biomarkers, right? Because I think what Chris just laid out there was perfect from a standpoint of how, you know, how we make the sausage, if you will, right? What we need to be able to have a legitimate conversation, scientific and regulatory conversation around the utility of these biomarkers.
Right. We recently, for instance, we recently did an exercise, certainly in the IBD community, there's a lot of interest in fecal calprotectin, and it's been collected by numerous programs for many, many years. And so when we looked at the data that was included as part of NDAs and BLAs over a period of time, you'd be surprised how many of those proposals didn't include units, didn't include any information around the analytics, and didn't include information about when and how the stool samples were being collected with respect to the ileocolonoscopy. And so if you're trying to make an association or comparison between fecal calprotectin levels and what you're seeing on the endoscopy, in the absence of that, it's really hard to make any comparisons. And so, you know, I understand that companies collect information for particular goals, but if you think of those goals in the context of the goals of the community at large, I think there's a real opportunity for folks to collect data slightly differently to benefit not only themselves, but also the community.
Yeah, Chris, I mean, we did a similar survey as well, right, within our pre-consortium. And what we found is that even though that data wasn't reported that way, those data are out there, right? So if we wanted to do something with fecal calprotectin this moment, we could do that. It's just there doesn't seem to be the interest from the community around that. Instead, there seems to be a lot more interest in basically these exploratory biomarkers, which I have to admit are a heck of a lot more exciting from a mechanistic standpoint and potentially from a predictive standpoint. But we have to start somewhere, and I think that has to be a part of the conversation, right? How do we build our knowledge? I really like what Brian said from the standpoint, he brought up, you know, what we understand about the current standards, right? That could also be a place to start, right? So those are the things that we need to think about as a community. Any other comments there?
I think that's the most legitimate request from patients to find a surrogate biomarker or surrogate endpoint that would replace the endoscopy. So if fecal calprotectin is the best candidate, I think all work should be focused on, not all work, but I mean, whatever is available to answer the question. Because for the moment, the situation is that fecal calprotectin or any other biomarker cannot be accepted as surrogate biomarker, surrogate endpoint. Hopefully,
Michael, let me just throw in a challenge here, right? Because, you know, academicians, their careers are all about publications, right? And they need to move on in their careers. They're not going to take time and do exactly what you just said, right? That's why these public-private partnerships and these interventions should come in to really take that step. I think that's a gap. You know, I can't imagine Parambir is going to stop everything and just work on this, you know, because this is not his career. Right.
But I think, Vijay, I think pharmaceutical firms have basically collected that data, right? And I think what we could do is utilize a lot of that data to do exactly what we said. I mean, I haven't looked at the patient level of the data, but looking at the metadata that's available in the survey that we did,
Yeah.
I mean, that information's out there.
No, fair enough, except the pharmaceutical industries that are in the biologic space are not basically they're not labs. They're not selling biomarkers. That's not their business model, right? So
Right.
that is the real disconnect, right? So the pharma industry will publish their data set with CRP and response, but they're not going to take that extra step because that they're not selling the kit or what have you. And the academicians are not, they're also busy with their new kind of what we call exciting biomarkers that Parambir and Ashwin and Stefan are doing, and what Fred's doing in his team. I think they're looking for novel mechanisms and stuff like that. But there is that as you said, there is still work to be done to take it to the next step. And when you talk to Chris Tarrant and Richard, this is what it seems like we need to do those things.
But isn't it compelling if we could decrease the number of colonoscopies that are required for a clinical trial? I mean, would that be a positive thing? I mean, especially in the pediatric space where sometimes that data is simply not accessible.
Yeah, right. Fair point.
And so I think that's what I think we're talking about. Nobody's going to make money off of this per se, right? We're not talking about that. But what we're talking about is creating a regulatory pathway that makes it easier for folks like you, Vijay, to get your drugs out there to patients in need, right? That's what we're talking about. And that's where the investment is.
Right, and I think also it may not be that they have a kit to sell, but if we were to find a biomarker that is a good indicator of disease activity so that we don't have to do ileal colonoscopies and trials, while you may not be making money from a kit that you're selling, you're saving money from not having that additional expense in your trials.
Exactly.
Exactly.
Well said, Chris.
So
Michael, Tara, I know that we talked. Oh, yeah, please.
Yeah, no, I think these are all great points, and what we're trying to do in 3TR, we're trying to bring the community together. That's why we started the work with the c-path and started this discussion so that we can kind of really aim to create a blueprint for what do we need to do for those exciting biomarkers to bring them through, right? But then I think we can also look outside of the current framework and say, what can we do to make a difference and to utilize the data that are out there?
Yeah, I think it's a really good point. And I think what it is, and I'll say this for a little bit of a laugh, but to be truthful, all of the biomarkers that are going to have impact in the near term on drug development are the boring biomarkers, right? From a regulatory decision-making standpoint. For internal decision-making, you're right. I mean, some of these more exploratory novel biomarkers may be able to drive, the process internally. But I don't think that we have the data set to be able to convince ourselves or regulators that we can make regulatory decisions around those biomarkers yet, right? But we have to create some type of baseline or foundation for us to be able to take that next step off of, right? Or else we're just going to keep swirling, right? And that's what I'm afraid of.
Well, and John Michael, you bring up a good point in that ultimately there's a lot of interest in different community groups in this space. And perhaps they have some overlapping goals. Some of them may be unique. But I think if there's a forum where folks can come together and identify and prioritize, say, their top 3 objectives, and then work together to figure out what can they contribute towards those. I'll use the NASH space as an example. The NASH space, identified early on when you're trying to develop clinical trials in that space, liver biopsy is part of how you determine if you're going to have the appropriate histology to support entry into a clinical trial. Everybody agreed in that community, liver biopsy, given its invasiveness and some of the safety concerns, if we can find a non-invasive way to assess the activity to determine who would be appropriate to enroll, that would be a worthwhile objective that everybody could rally around. And what we've seen in the biomarker qualification space, I think we're now up to 10 plus different proposals from different groups, basically targeting that very concept of looking at both imaging and non-imaging biomarkers trying to identify the likelihood that someone's going to be biopsy positive. That doesn't mean that we're replacing the biopsy. That is the ultimate goal, but it's a step towards reducing the number of biopsies that you have to do to find the right patient population. I think in the IBD community, if the community as a whole could identify, what are those key stumbling blocks, both for trial development and then also for patient care, and especially if those two overlap, that would be a good target to see what's out there from a biomarker point of view.
Perfect, Chris. And we do have a good start at that, right? We did pull folks together to do that. That's what our IBD, or I'm sorry, our Crohn's biomarker review is about, right? It's really trying to ask the question about what are the unmet needs, right, around this disease, and then what are the potential solutions from a biomarker standpoint.
Yeah.
Right.
That's great. I mean, that's where we need to go. Any additional comments on this subject? Because I would like to talk a little bit about the how to do this step next.
John Michael, there's also a question in the box. So one last point about the calprotectin is, like, unlike albumin that was measured by Paramveer, calprotectin it's not a trivial way. It has challenges, and that's where some of the industry to real world has been a disconnect from a local lab to a central lab. I don't need to discuss this, but just throwing it out there just to complete the discussion.
No, I completely agree. And I wasn't trying to say that, hey, fecal calprotectin is what we should all be rallying around. In fact, what we found internally is that there's a lot of problems, which you just alluded to,
right.
That may, there's a good chance they can't be overcome. So.
And I would second that. Also, I didn't mean that fecal calprotectin needs to be the one, and there is perhaps a lot of room for scientists to make a career proving that there is a better one.
Right. The dipstick microbiome. Yeah.
So there is a question in the box. Thanks for pointing that out, Vijay. I've gotten carried away with this conversation. So it's from Gerard from Crohn's and Colitis Foundation. And he's speaking to the fact that there's a lot of variability in how we define treatment response within IBD, both for ulcerative colitis as well as for Crohn's disease. And that is there an opportunity to address this?
Yeah, so actually this is a 3TR question. So Fred, you want to take, I could take that too if you like.
I can start, Vijay. Yeah, we totally agree. There's a lot of variability in how we define response. And the endpoints are variable, and that doesn't help actually the cross comparison across studies in defining biomarkers. I think the other issue is the one I already highlighted, is the inherent imprecision in many of those endpoints, which is embedded in how the scores are set up. So totally agree with your point.
The only thing I would add, Gerard, is we're trying to ask the following question. So we're letting the clinicians within their spaces work independent of the 3TR. So if Stefan says the patient is in remission, as long as all IBD folks are using that same exact tool, we're okay with that endpoint. So from a biomarker perspective, we're saying, what are some of the parameters that gives us the super responders and super non-responders for every disease subset based on their clinical thought leaders and their endpoints. And then when we analyze it, then we're going to try to come in the middle to fill the gap. I mean, that's sort of how we're trying to approach it.
Vijay, you bring up an excellent point. I mean, what we found, I'll use my NASH example again, and then also traumatic brain injury, that you needed to have consensus definitions around who the patient is, and if it's a chronic condition and there's sort of a waxing and waning character to it, how are we going to define disease activity and severity? And once you have those consensus definitions and you share them with the community and the community then adopts them, it kind of gets to the issue that you're raising. But if you're allowing physicians and groups to sort of come up with their own favorite definition, you don't really have a hope of trying to gather information from multiple sources and trying to make any sense of it.
Thanks.
And this has been a focus of Crohn's and Colitis Foundation and their biomarker summit that they've been holding. We also know that the Foundation for the National Institute of Health is trying to come up with some consensus definitions around endoscopic healing, right, at least for ulcerative colitis. And so there are these activities going on, and I think that's one of the difficult pieces, right? To be able to understand the landscape in a broad enough way to be able to pull all these ideas together, right? Because we can't work independently. We know that, right? And so even public-private partnerships, consortia, need to collaborate in order to get these activities done in a reasonable amount of time. Okay, great conversation. So I think one thing that would be interesting to discuss is as we begin to think about bringing these biomarkers forward for regulatory endorsement is a little bit around the differences between FDA and EMA in how this process works. Chris, you gave an excellent presentation around the biomarker qualification program at FDA. Ricard, we didn't have an opportunity to get that same presentation from EMA, but can we compare and contrast some of the differences? Because it looks like the process is different, and so some of the scientific thinking and expectations.
Yeah, I mean, I can start, or Ricard, you can. I don't really, doesn't matter. But just some overarching comments. One is that at least EMA and FDA both have qualification efforts, and they have had for many years. We have monthly conversations between the two groups where we talk about policy and process issues and trying to achieve alignment, but then we also talk about projects. So if a group is engaging with both of us in parallel, there are opportunities to sort of have us invited to each other's meetings as observers. And then likewise, when we are developing our recommendations, we try to share them with each other to again, achieve alignment when feasible and possible. One of the challenges is that I think we had a great more alignment around process pre-cures, but then when the cures legislation came along and defined a three-step process, that's where we diverged a little bit. I don't think it's insurmountable in that the previous process, you kind of start with the letter of intent, you had a number of briefing packages, and then you eventually got to a full qualification package. The real benefit of the qualification plan is that it puts in writing and clearly defines, what remains to be done and what data would be collected and how it would be analyzed so that there's not a moving target. And EMA, although they don't have the equivalent of a qualification plan, the concept is there, and they're very much open to that. So I don't see the process per se as being the difficulty. I think it's more around aligning of what the objectives are. And that's probably a bit more of a hurdle.
Ricard, you're on mute, I'm afraid.
Yes, sorry. I was unmuted when I didn't need to be. So yeah, I think there are not really tremendous differences in the objective of the process. Maybe what I would like to mention here is that we have this ITF interaction, which is Innovative Task Force, which is free to any consortium or any industry, any person. And it is actually the first place where the concepts already can be discussed without any binding from any side. And that is kind of a filter or advice whether the concept already is applicable to regulatory needs. And I would recommend really to come early to that one. As for the qualification procedure itself, depending on the outcome, we offer basically two things, either advice or opinion. Opinion is something conclusive on the acceptance of the of the tool, in this case biomarker, and that is then published and available for the external world. When there are things to improve or amend or develop further, then we don't accept opinion, but just an advice, which is then not public. There is only a summary published in agreement with applicant if needed, but that is then perceived as kind of ongoing procedure, ongoing rounds of interaction leading finally to the opinion on the biomarker method or whatever. So that is maybe a difference, but basically, as I say, I don't see too much differences in objectives. And maybe it would be good to hear from participants here whether they perceive any significant differences, or whether they, because we feel that the uptake of the procedure, is not that great as we expected, and maybe there is something that we should improve to really offer this because it's time consuming. It's quite difficult procedure. So to invest there, we really need to understand what is maybe expected to be better.
Since everybody else made a plug for their respective organizations, one of the things I do want to bring to your attention, at least for the next couple of years, is that if you have an accepted LOI in our program, you then become eligible for a Ddt Grant that can be up to $250,000. It doesn't mean that you're guaranteed a grant if you're in our program, but you are at least eligible for that. And especially if you're doing some type of a collaborative effort and working with academics and even for pharma partners, that additional funding can be very helpful as part of your development effort.
Thanks, Chris, for bringing that up. And so c-path has received some of these grants, and they've been very helpful, and it was quite a collaborative mechanism to receive that grant. You know, as someone who is a requester or a DDT applicant or a novel methodology applicant, I mean, I perceive there are some differences between, FDA and EMA, not only in the process, right, but also in some of the thinking around what the regulatory or data expectations are. And so that's why, what we like to do is engage both FDA and EMA, because we know that drug companies are going to be using these biomarkers worldwide. And so we want to make sure that in the end, we at least have a context of use that's similar, right? That can work together, and that we're able to achieve, recognition or endorsement from the major health authorities that yes, this biomarker can be used. So I think that has to happen, and that needs to be in everyone's mind, that it's very valuable to engage both regulatory agencies and possibly even PMDA, depending on what your strategy is.
So, John Michael, there was a question in the chat box from Stefan Schreiber. If understand, they wanted to know more about the grants process and the submission path. So just know that you have to have a letter of intent first. So if you look at our website, it certainly gives you details of how you submit a letter of intent to us. Once you're in our program, namely that you have an accepted letter of intent, we then do outreach to eligible partners and alert them to the grants process. But the first step is that you have to have an accepted letter of intent.
Perfect. Thanks, Chris. Let's also socialize Ricard's question around, are there perceived barriers or are there perceived differences between the two agencies, from any of the panelists on what your thoughts are around that? Vijay, any thoughts there?
You should ask Pam and Elizabeth, and what do you guys think from a regulatory perspective?
I do believe. Oh, Pam, would you like to go first? Go ahead.
Go ahead, Elizabeth, please.
So I do believe Richard brings up an excellent point. The acceptance of the qualification pathway with EMA has not been probably as great as one could hope. And the complexity behind the pathway is probably, or the mystery behind the pathway is what makes it not so widely used, because what it almost intuitively, what we have to do, we have to go for the innovative task force meetings as in our letter of intent equivalent in Europe to start this discussion, because the qualification pathway is really once you've made up a plan. So therefore, bringing these two together and bringing the innovative task force discussion in as a kind of add-on part is what we've been doing across other EMI initiatives as well as we proceed through this process. And I think the perceived hurdle is quite high, but that's again because there is no initial distinction. So you can either have an opinion, if I understand it correctly, or an advice. But going through the advice procedure, it's not always clear as to what can you expect as an outcome.
Pam, do you have any comments there?
Yeah, thanks for the opportunity to comment. I think, you know, as you think about the various what needs to be done to move through the processes, I think it's really aligning on the context of use. I think you've touched on it yourself a couple of times, John Michael. That's really critical. And while we understand that, you know, looking, for example, at the US procedure, you know, the data needs around the letter of intent are modest, as has been discussed. So, you know, kind of entering into that process of the letter of intent is not reliant on extensive data, but it's really conceptually, right, aligning on that context of use and doing that in a really forward-thinking way. I think is really critical to success. And you know, whether you're achieving it through this, you know, through the letter of intent, or whether you're achieving it through the process that Elizabeth just outlined, that's really where I think the harmonization needs to be. Because if it's divergent at that point, then it's going to be divergent through the entire process.
Right, and John Michael, that's what I was referring to is, you know, the objectives. It's not necessarily that the objectives of the two qualification programs and the two regulatory bodies are different. It's the objectives of the requester who's ultimately trying to put forward their project proposal. And Pam, you hit the nail on the head. The context of use is what's critical. And what we really spend a lot of time doing in those early engagements is really trying to critique what it is that you think, if this were qualified, it would do. Many people come in with sort of multiple objectives, all kind of crammed into a single context of use, which makes the first thing we do is sort of unpackage that so we can focus on a single one at a time, so that our recommendations are going to be appropriately specific. And likewise, you need to define your context of use within the current paradigm. A number of times people, when they discuss a drug development need, really don't describe, well, what is the issue in a clinical trial as it's currently run, and then how would your biomarker benefit if it were to be qualified? You really have to kind of do that level of specificity. An additional comment related to sort of engagement, at least on the FDA side, we've seen a tremendous increase in the level of engagement across all parties, academic, pharma, and others, especially since the cures legislation. Pre-cures, the program was receiving maybe 2 or 4 letters of intent a year on average. Pre-COVID and post-cures, we were seeing 2 to 3 proposals a month. And so the level of interest has greatly increased. And the level of engagement with us by academics has also increased, and the quality of the proposals has increased as well. So it seems that, at least for us, the Cures has really done a tremendous job of creating awareness, and thankfully the transparency provisions are making it easier for folks to better understand what is asked of them at the different stages of the process.
Yeah, thanks, Chris. And I think one important aspect to remember too is that, you know, this interaction with the agency is very different. Both agencies, both EMA, FDA, and PMDA, is very different from you know, the IND drug application process. It is a specialized area of both regulatory science and regulatory affairs. And I think that's part of the problem, because when you start to talk about this, it feels so much like drug development. I mean, that's what I did before I came to c-path. And it does. We use similar words. We go ahead and talk about it in similar ways, but it's very, very different in how it ultimately needs to be presented. And the strategies behind the programs are very, very different. And so that's something to keep in mind. And again, you know, that's what we specialize at Critical Path, right? Is to be able to have these types of interactions and get programs positioned such that we're going to get the outcome that we want. Any other comments around that? Because we're about to go into our speed round of wrap up in about 2 minutes. So. Okay, there is one thing that I want to bring up, and I saw this in the chat box that we really didn't address, and that was around the idea of AI and black box, right? And this is more a question to health authorities. I mean, clearly, you know, there's less resistance in, say, clinical practice to be able to use something that will give you the outcome that you want without having a complete understanding of how that algorithm or that approach is giving it to you. You know, Chris, Ricard, Tara, you know, is that okay for, you know, an FDA or EMA qualification submission, or do you really need to know what's going on in that black box?
No, I mean, we're open to any and all technologies, and certainly artificial intelligence and machine reads. We have examples of those that are in our program at present. Ultimately, there's different ways you can use AI. AI can be a learning process to ultimately define a signature that then is static. That sort of approach is probably one that we're more comfortable with as regulators. We don't really care how the AI worked to get to that ultimate algorithm, if you will, as long as that algorithm can sort of be shared and be shown to be reproducible and reliable. However, if you're talking about AI and that it's a system that's always learning and therefore always changing, that gets to be a bit more problematic in a clinical trial context, especially when clinical trials are conducted over multiple years. And so if the tool in one instance isn't the same as the tool for other patients that are ultimately going to be included as part of a data analysis, then it becomes difficult to think about how the data could be interpreted.
Thanks, Chris. Any comments, Ricard, Tara, before we move to the speed round?
I can only agree with Chris. Of course, it's good to understand what is in the black box, but we cannot pretend that we always understand also any mechanism of action of drugs which we approve frequently. So yeah, it's only important whether it works and whether it's reproducible and what results it brings. So yeah, we have to agree. We have to live with the situation. We don't understand everything.
Great, thank you. Okay, so why don't we go ahead and complete this panel and go ahead and wrap up. You know, first of all, I'd like to thank the panelists. I think we had a great discussion, and I think we went in some, you know, really positive directions. So why don't we start to wrap up? Tara, could you go ahead and give some closing comments and begin the wrap-up process?
Sure, I'd be happy to. So, you know, it was great to listen to all of the discussion, and thanks for all the great questions. The one point that I wanted to make that I didn't hear discussed that I'll leave you with as a thought is just that, you know, I heard multiple people allude to the value that we would all see coming from replacing colonoscopy. And I think that that's a laudable goal. It's certainly in the best interest of patients, potential trial participants, and, you know, as well as companies and regulators for the reasons that people mentioned in terms of it might be less expensive and easier to recruit and things like that. But I also think that that's a really high bar based on where we are today. And so I hope that throughout the discussions, all the different stakeholders have heard an interest in developing things somewhere as an intermediate step. I think, at times you get proposals for some type of novel biomarker to replace colonoscopy, and for various reasons, based around the complexity of the disease and what we're trying to measure, we're not always able to get on board and say, yes, that sounds like a great plan and we can get there in 2 years. But I do think that there is a lot of value to exploring different response biomarkers, different predictive biomarkers, different modalities, including imaging, that could help us in our shared goal of getting there eventually. And so I really hope that those different types of opportunities came across in the discussions today, and that people will leave here with some appreciation that we do see value in those intermediate steps. It doesn't have to be an all or nothing prospect where if your biomarker isn't going to get you out of doing colonoscopy, then it's not worth your time, because I don't think that that's the case. I think that was the only other thing I wanted to add. So thanks very much.
Great closing point. Thank you. Elizabeth?
I'll take myself off mute. I think one of my takeaways is that a lot remains to be done. We've in the beginning of the journey in this space, and I think 3TR, c-path, and the company's involvement and the academics' involvement and the patient involvement highlights the need and the gap that we do have, and that we should find a way to address it, hopefully sooner than later. But just like Tara said, we should have the realistic goals to achieve throughout the process. And thank you very much for organizing, obviously.
Thanks, Elizabeth. Fred and your internationally famous cat?
I will take home actually what we're doing today, which is the bottom line, which is to interact with the health authorities, whether it's FDA or EMA, quickly, early in the process and often. So I think what we're doing today is a great example of that, and I look forward to continue that conversation. I agree that what we're trying to do is ambitious and difficult, having been in this business for a while. So again, I look forward to our next conversations.
Thank you, Fred. Pam, do you have some closing statements you'd like to offer?
Yeah, thank you. I think the emerging consensus is it takes a village. And that takes us back to your concept of community. And I also like the, the idea that we're getting there together. It's going to take us a while to get there, and that we should not shy away from modest goals along the way. And I take on board as well, John Michael. I think at one point you said, it's probably the boring biomarkers that are going to emerge first, and it's probably true, and I think that's because of the depth and breadth of information and data that's necessary. It just takes time to develop those data sets, but each step is progress. Each step is progress, and we shouldn't be deterred because it takes time and it's difficult. I think that's why we all do what we do. It does take time. It is difficult. Yeah, thank you for the opportunity to participate.
Thanks, Pam. Chris, closing statements?
Yeah, I would kind of summarize what many of you have commented on in a slightly different way. I would encourage people to actually sit out at a whiteboard, put out your roadmap, all the different components of what you think the field needs. And then for each component, put together sort of a set of milestones and needs. And then look really critically at your resources, and resources include financial, expertise, time, etc., and match your resources to an objective. I think many times what we see is that people choose objectives for which they're not resourced to achieve. And that gets to be a bit problematic. So it again gets to the earlier points of It may not be a sexy project, but if it gets you towards the goal that you're trying to get to, then it's a worthwhile enterprise.
Thanks, Chris. Ricard?
Yeah, thank you very much for putting this together, John Michael and colleagues. I think it was very nice, and I would give a parallel here to what I'm now working on for COVID-19. There are a number of studies that don't think about regulatory implications, and they are in the end not usable for regulators. So that's an important dimension here. I think that these researchers who are developing biomarkers need to think about regulatory impact of their work, whether they are usable. Thank you again for putting this together. I'm looking forward for further interactions, not only because they are important, but of course also because they are not boring, they are very interesting. Thank you.
Thank you, Ricard. Thank you, everyone, for your words of wisdom in the wrap up. Vijay, would you like to wrap this session up as well?
Well, I want to just thank everybody. I want to just, I learned so much today, right? And really be honest. And to me, what the most important thing I learned was, you know what, I was still hung up on these numbers of a positive predictive value in the clinic and a negative predictive value in clinical development. I think that's a really nice message to take and anchor and try to develop the biomarkers through all these different paths. I think the patient voice is very powerful, extremely motivating. And as you know, the future of those gene expressions and microbiome, I think that we have a really exciting path forward. So thank you once again.
Thanks, Vijay. So why don't we close things up overall? Chandler, if you want to go to the next slide. I think we did, make some movement on this goal and these objectives, right? I think we had some really good conversations, and I think we all learned a lot about this process, right? And again, to repeat one thing that I think is really important is that to retrofit a project to go ahead and have regulatory goals as an outcome is very, very difficult. Those regulatory goals need to be upfront. I think that was a primary learning from today's conversation. Then likewise, I really liked Chris's idea that he brought up and that others, also rallied around about pulling together these different ideas, right, and deciding as a community what we need to invest our time and money on, right? That's the important part. What's going to get us the biggest impact in the soonest amount of time that we can then build on. Personally, I loved where the first panel went as well. I mean, I thought we talked a lot of good aspects to not only drug development, but then also the clinical practice, and then understanding, from Bing what the patient really needs, right, and what they're looking at. And that represents the community, right? Those are the conversations we need to have, but we also need to have the discipline to be focused on what our goals are, right? Because you can hear in some of the conversations, and I warned you about that it kind of matriculates between drug development and clinical practice, both very important, but in some ways, the expectations are very much mutually exclusive, right? For what types of data needs to be produced and how that data needs to be presented in order to achieve those biomarker goals. Before I turn it over to Vijay to wrap everything up, I'd like to thank not only the participants today, but also the planning committee, where we had multiple early meetings working through this, trying to get this to resonate the right way, and then also to the c-path staff for working the operations. I think everything went really well today. I was quite happy. I mean, a couple little technology hitches, but hey, we expected that. So thanks, guys. Really appreciate it. Vijay, I will give you the last word.
Thank you so much. And, you know, we spent 4 hours, you know, I know everybody's very busy. So from the bottom of my heart, thank you, everybody. And thank you to the participants, you know, whose names were not seen, but, you know, without them, this is nothing, right? So hats off to all of you, and please stay in touch. Let's try to do this once again at some other time.
Great. Well, thanks a lot, VJ. Let's go ahead and break it. Bye-bye, everybody.
Bye. Bye.
Talk to you soon.